This video examined how we can use an evolutionary approach to better understand issues associated with HIV. HIV is actually a group of viruses called the human immunodeficiency viruses, and it results in an illness called AIDS, which is acquired immune deficiency syndrome, and this is actually wrapped up because of the biology of the virus and its reproduction. So what I want to do in this video is give you a better understanding of the evolution of HIV and how our understanding of its evolution can help to address several health-related questions in humans. So the first question we'll address is, why do some drug therapies work well initially, but then they stop working? And how we've used this information to improve drug therapies to improve the long-term prognosis for individuals affected by this virus. We'll also look at the question of, why does HIV kill people? How is that in the best interest of the virus's evolution? We'll then address the question, why do some people show some natural resistance? And what does that mean to future populations? And then we'll also look at how we can use phylogenetics to understand the origin of HIV, or actually we'll see the origins, and how we can actually use phylogenetics to understand patient transmission. Let's first just talk a little bit about the epidemiology of the virus and the virus's biology. And I have to admit that these stats are a little dated because your textbook is just about up for a new edition. But I think it's going to get the point across. So at the time your book was written, there were 76 million total people infected worldwide, and that's resulted in about 35 million deaths thus far. So that's a huge fraction of the total number infected. In 2016 alone, there were 1 million deaths attributed to AIDS. That is sadly a high number, but the good news is AIDS-related deaths have been falling since the peak in 2005. So the 1 million deaths in 2016 is actually a reduction compared to what we were seeing in the early 2000s. If you look at the demographics of the virus, you see that in the US, the overall rate of infection is 0.6% in the population overall, and it seems to be concentrated in homosexual men, which represent about 8 to 24% of infections, and IV drug users, which is around 16%. We'll talk about some other breakdowns of the remaining individuals in a bit. But you can see by looking at these bars, there is some variation across the globe with regard to the fraction of women, men, and children that are infected by the virus. And on the x-axis, we're just showing you that there is still an increase in most populations with the total number infected. You can see in South Africa, there have been steady increases in life expectancy until fairly recently, and this may be attributed to an increased abundance of individuals infected with HIV that have led to a decrease in life expectancy. So you can see at about this same time period, these are not on the same scale, but about the same time period you see the peak of infections in South Africa. We see a decline in life expectancy. The most common modes of transmission seem to vary among populations. You can see that by looking at these pie charts here. For example, in Kenya, look at how much of this is due to the sex trade. In other areas, there's a large fraction of this associated with heterosexual sex with a high-risk partner. Casual heterosexual sex is also a big issue in Kenya, Honduras. In Russia and Indonesia, injection of drug users, IV drug users, is the biggest problem area. And so there's going to be needed a different social approach in different countries to combat these transmission rates. Just looking at the US, Canada, and the UK, a different study showed that, again, homosexual men have the largest fraction, but look at the high fraction of heterosexual sex. So I'm just urging everyone not to add a stereotype associated with that's the only way you can get it, either through homosexual sex or IV drug use. Heterosexual sex is a major transmission mode. And while there was a large number of infections in the mid-'80s, there was a steady decline associated with education, an increased use of clean needles and condoms, and just safer sex in general. But we're going to see that there has been the production of more efficient drug therapies, and unfortunately the new infection rate is increasing in recent years because people think, it's no big deal if I get it. There are medications for that now, but we'll talk later about why that's not the best approach, not the best attitude to have. All right, a little bit about the virus itself. Viruses are non-cellular infectious particles. There's a debate among biologists whether they're actually alive or not. They have certain characteristics of life forms, but in other ways, they are not life forms. One characteristic of all life forms is the ability to reproduce. And yes, viruses can reproduce, but they're incapable of independent reproduction. The viruses have to invade a target cell, in which they then hijack the cellular mechanisms associated with normal reproduction of the host cell, but they use those mechanisms to make copies of their own viral components. So this means making the viral DNA or RNA, depending on the virus type. HIV happens to be an RNA virus, and then the protein coat that surrounds that genetic material. As I mentioned, HIV is an RNA virus. It's what we refer to as a retrovirus. This means that a virion, one of the viruses, has a surface protein that has to match the co-receptor in the cell membrane of the host cell so that it knows which host cell to invade. Once it makes that recognition, it then injects its RNA into the host cell. At that point, we have reverse transcriptase, turning the viral RNA into DNA, which then is spliced into the host DNA, and then the host produces, through gene expression, more RNA copies, messenger RNA copies of the viral genes, which then are expressed to produce the protein coat and make a new virus, which is put back into the bloodstream. So different viruses have different effects on the host's health. In the case of HIV, again, there are serious consequences of this. It causes AIDS, acquired immune deficiency syndrome. And that is because the HIV virus attacks immune cells, and this destroys the immune system. So people don't die of the virus itself, they die of opportunistic bacterial or fungal infections that they would have normally been able to defend against, but their immune system has been compromised because the immune cells have been killed by the viral reproductive capacity. So if you look at kind of the life history of the virus in a typical patient, you see that there's an acute phase immediately after infection where the viral copies are at a very high rate and it is negatively impacting the immune system. So you see that as that is going up, the helper T cell count drops drastically because these cells are being killed by the virus. Then the viral load is actually decreasing for a bit, and it goes to a chronic phase in which it's a relatively low concentration in the blood supply. But if you look at how that's related to the production of T cells, immune fighting cells, they have an initial rebound, but then they go through this slow decrease as the HIV virus that is in the body continues to infect them. And once you end up having the T cells at such a low number, then you have this rapid entry into acquired immune deficiency syndrome, AIDS, where your immune system collapses and you're more likely to be killed by an opportunistic infection. So some of the target cells initially are associated with the gut tissue. So as you get the HIV virus in the acute phase, it depletes your T cell helper count. This impairs the gut's defenses initially and allows for bacteria and fungi to bypass the gut tissue and get into the bloodstream. Now what that does is it induces, once those pathogens get into the bloodstream, the immune system kicks in and it says, okay, we've got a problem here, we got to start dealing with this. So we get the B cells produce, and then the T cells. But remember, HIV is beginning to attack these as these are going up to fight the bacterial or fungal infection. That's producing more target cells, the immune cells, that then the virus is able to attach to and make more copies of themselves. And so it's this negative cascading effect. The sicker you get, the greater your potential immune system response, at least initially, until it's just overwhelmed by viral attacks that kill off those T cells and exhaust the body's ability to make more T cells. Well, some of the earliest treatments associated with HIV included the use of a drug called AZT. What AZT did as a proximate mechanism, how it worked, is it stopped that process of reverse transcription. So as the viral RNA was turning itself into DNA that would then be incorporated into the host DNA, the drug was mimicking thymine. And so as the reverse transcription was going through, we have this polymerase that's grabbing the nucleotides, putting them together based upon the pairing rules. If it grabbed an AZT instead of a thymidine, this would actually stop the process of reverse transcription. It would therefore prevent the viral production of its RNA signal into DNA signal, and then it couldn't be incorporated into the host DNA system, and it stopped additional viral replication. So this had some promise early on, but it only worked for a few years. Not only that, it did have some side effects to the patient even when it was working. But let's really focus on why it stopped working, why it worked initially, and why it stopped working. Well, HIV is the fastest known mutating organism ever. If you look at gene sequences before and after a treatment, you see that the viral strains that were sampled initially have significant genetic changes as they remain inside of the patient. And this is because of mutations that are occurring during reverse transcription. The reverse transcription process is very faulty, and it oftentimes produces DNA that will then translate into messenger RNA, viral messenger RNA, that accumulates mutations. This is one of the reasons why this was a target of initial drugs, trying to take advantage of how sloppy these polymerases were and non-selective. The result is that about 50% of all reverse transcripts have a mutation. So it has a high mutation rate, and the other thing is it has just a high reproduction rate. And that means it's very quick generation time. And remember, evolution works at the level of a generation time for the organism involved. So the faster the generation time, the faster that organism can show adaptive evolution. So at this point, we're just talking about it accumulating mutations. Well, what could those mutations do? Again, what you see is that after months of therapy, you see a rapid rise in the resistance to the AZT treatments. And at that point, the drug is just not effective at all. See, what has happened is some of the mutations that have occurred have actually produced a polymerase that works during reverse transcription that is just more efficient. It's more selective, and it only picks up the thymidine and ignores the AZT. And once a lineage of viruses get that, it will sweep to fixation in the patient. Here's a graphic that basically shows that. We start off with viruses that are susceptible to AZT. If you get a mutation that makes one a little bit resistant, it's going to increase in frequency compared to the original strain, which is eventually going to go extinct. It may persist longer, but if it also gets a mutation that is highly resistant to AZT, then that is going to sweep to fixation in the population. So the drug may work at first on the non-adaptive strains of HIV, but if you get an adaptive strain, what we're showing is natural selection has produced a strain that has better differential survival and reproduction. It's an adaptation from the virus's point of view. And what we see is this happens repeatedly in different patients, that these strains are independently accumulating the same mutations that give them AZT resistance. And this is because of their fast generation time and their fast mutation rate. So they're converging on the same adaptation in this coevolutionary arms race between the virus trying to survive and leave copies of itself and us trying to keep the patient alive by stopping the viral reproduction. It has been shown, though, that if you take a patient off of AZT, that these AZT resistant strains decrease, which indicates that without that selection pressure, there is a cost to producing the polymerases that are pickier, so that when that selection pressure is reduced, we see the evolution again of viral strains that are less resistant. Well, understanding that this happens has led to the development of a strategy that beats the evolution of resistant strains. The idea is to develop a drug cocktail that has multiple drugs. See, if each of the drugs targets a different life stage of the virus, a different mechanism of the virus, a single mutant virion may have resistance to, say, AZT, but it may not have resistance to the other drug. It makes it more difficult for the virus to evolve via random mutations in a single lineage, mutations that defeat all, say, 3 or 4 drugs. And sure enough, these drug cocktails are shown to be more effective in suppressing viral loads. So HART is one of these treatments, and we see that compared to the pre-HART survival rates, you see that there is steep declines in survival prior to these drug cocktail treatments. Some of the earlier drugs used provided significant help. Modifications of these drugs in both which drugs and which concentrations has led to even a greater protection. However, I want to make it clear, while these are beneficial, this still doesn't compare to the annual survival at different stages of people that are HIV free. So yes, these drug cocktails are helpful, they do help, but the current drugs can't completely eliminate the virions. They can make it to such levels that it reduces transmission and it reduces your likelihood of developing AIDS, but they're not perfect. And again, with all drugs, you can have side effects, and remaining on a long-term therapy, having to take these drugs your entire life is not preferable, both from a physiological point of view and then also just the cost associated with it. Well, it turns out that there are some people that appear to have some resistance to HIV. And one of the alleles linked to this resistance is at the gene CCR5. This produces a co-receptor on the cell surface of these immune cells. And if you have the allele called delta 32, it reduces the chance the virus will recognize a cell and be able to successfully invade. Now, not all populations have the same frequency of this allele. It appears to be higher in Northern Europe and rarer in Asian and African populations. So why is that? It's possible that this is an exaptation for HIV, and that this delta 32 has evolved its original adaptive function in a different context. It's been suggested that it may have helped with the bubonic plague, which was a severe force that killed about a third of Europeans in the 14th century. It may have also been linked to greater survival possibilities associated with smallpox. So it could be due to past selection, and that this is really just a reflection of what we sometimes refer to as the ghost of selection past. It's maintained in the population just because it was at higher frequencies in the past, and it's just kind of being held over through inertia. It may be also possible that it's spread throughout Europe due to gene flow because of the high frequency of Viking invasions in European populations. And in some of these populations, genetic drift may have increased the frequency of this virus just by sampling error due to genetic drift in small populations. But regardless, this isn't too surprising. Similar alleles have been found to confer resistance to SIV in different monkey species. SIVs are viruses in the same family, very closely related to HIV, and so studies of those have helped us understand, again, HIV's properties. Another resistant allele is the CD4 cell receptor, the genes associated with that. In a study in Kenya looking at female prostitutes showed that certain genotypes allowed for individuals to stay in that industry longer before they got infected. So while we're trying to work on drug therapies to reduce the likelihood of getting or living longer with HIV, there may be certain genotypes that could evolve in populations.
Now let's look at how we can use phylogenetics to see where HIV came from. So here are two studies that looked at the relatedness of HIV to closely related SIV viruses, and we can learn a couple of things from these studies. The first is using what we call a molecular clock by estimating the mutation rate and the evolutionary rate of each of these viral lineages. We can see when they all share a common ancestry. Using this technique, we're able to see that HIVs have evolved in the time frame of about the 1930s in human populations. The other thing is, HIV is not a single virus. HIVs, different strains of this virus, have evolved independently a number of times from different SIV lineages. So here's one case of it. Here's another independent case. Here are two more independent cases. Now we knew that these lineages down here and these did differ significantly in their nucleic acid sequences, That's why this is the HIV 1s are more similar to each other than the HIV 2s. But even the HIV 2s and the HIV 1s, there are multiple strains within those major types. So how did these get into human populations from SIV viruses in other monkeys? The most likely scenario is associated with humans hunting monkeys for food. When you're butchering animals, oftentimes you cut yourself, and so you're dealing with a bloody carcass, and if you cut yourself in a sharp bone or the knife that you're using, infection is fairly easy and likely. So the next topic I want to cover is the question, why is HIV fatal? Because if you think about it, the virus's evolutionary goal is the same as yours. Pass on as many copies of your genes as possible. And it requires our cells to make that happen. So why wouldn't HIV evolve to the point where it can live within its host without causing damage? Why does it cause host illness? Another way of putting this is, why is HIV so virulent? Virulency, we tend to think about that is how it affects the health of the host. A more virulent virus is going to cause more harm to the host. But really, virulency is a measure of the reproductive rate of the virus. And that makes sense, right? The virus is hijacking your own cells to make copies of itself, and as it does that, the faster it does that, the more of your host cells it's damaging, and so it's more likely to make you sick if it's more virulent, if it's reproducing more quickly. So virulence is related to the viral's reproductive rate. Well, from an evolutionary point of view, how would reproductive rate influence the fitness of a viral lineage? You have to think that there's selection for different viral lineages in the host immune system. So one of the things that's going to determine whether a viral lineage is successful or not is can it avoid the host immune system? Those that can avoid the host immune defenses more effectively are going to produce more copies. In this case, however, by successfully invading the host immune cells, since it's using the immune cells, it just happens to end up leading to a more rapid collapse of the immune system and an earlier development of AIDS. So a successful virus is better at invading the host immune system, but at the same time, that very success can lead to its ultimate demise within a single host. So its only hope is to be able to spread its virus from one host to the other. And see, selection for a faster reproducing virus in the host there would lead to an increased frequency of the virions in the blood. So if you're reproducing faster, there's going to be a higher concentration in the blood, which translates into a greater likelihood that you'll be able to transfer your viruses from one host to the next. But again, the flip side of this, if you make your host too sick, it could actually reduce the likelihood of transmission. If, for example, in HIV, if the virus makes you so sick that you can't have unprotected sex with someone, or you're unlikely to be in a situation to gather with other IV drug users and share a needle, again, that's actually a level of virulency that is too high for the good of the virus. On the other hand, if the virus reproduces too slowly and there's very little concentration of the virion in the blood supply, when an individual shares a needle or when an individual has unprotected sex, there's a chance that one, if it's a one and done kind of situation, then there's less likely to be transmission. So this means there's likely balancing selection on virus replication rates. And that's what this study here shows. If you look on the x-axis, we have viral load, so greater viral load over here. That's associated with a duration of AIDS-free infection years. You're not going to live very long if you have a virus that's reproducing at this rate. If the virus is reproducing at a much slower rate, it causes the individual to get AIDS at a much later time period. So it's less stressful, less damaging to the host immune system, and so they're not going to get these secondary infections. So we got that relationship. But again, the transmission rate, a higher viral load in the blood supply is associated with more likelihood of transmission infections per year compared to a low infection rate. So this means if you're going to look at the most likely transmission potential infections that is going to be the highest. It's an intermediate level of viral load, which means an intermediate virulency. So just to wrap up this section on HIV, what have we learned by taking an evolutionary perspective to look at HIV? Well, one, we figured out why single drug were ineffective long term, that there was resistance evolving in lineages, but how multi-drug therapies can be much more effective because a single virion lineage is unlikely to gather the appropriate mutations to counter multiple drugs. And unfortunately, this has led perhaps to some more risky behaviors in some high-risk populations. It's going to take some education to show why that's not in their best interest, despite the fact that these drugs are fairly effective these days. One interesting factor is that there is evidence that humans could gradually evolve resistance to this virus. Now, that'd be a long, painful process through natural selection, but it does show that there are alleles for resistance to these infections, and we already see that the pattern is seen in closely related primates that are dealing with closely related SIV viruses. So by studying these, it may help us understand the action of these alleles and why they cause resistance, and perhaps that will help us develop a vaccine. However, that's a tough task. Remember, the HIV strain, there's not a single one. It's a highly diverse set of viruses that show a great deal of genetic diversity because of their multiple evolutionary origins, and the fact that this virus evolves so quickly because it has such a high mutation rate. And these are going to be challenges for developing a vaccine. One other thing about using a phylogenetic approach related to HIV, I mentioned this study earlier in the semester. This is a situation where a doctor was charged with infecting a former lover who was actually a nurse with HIV-tainted blood. But to prove this, the prosecutor had to prove that the victim strain that she was infected with was more closely related to one of the doctor's patients than just some general individual in the population, because the defense was arguing that she could have picked this up anywhere. Well, again, we know how this virus evolves. And so in this graphic, it shows you if you have one patient that if they have it long enough, they're going to develop their own individual strains because of the high mutation rate associated with this virus. Well, if this individual infects another individual, it's going to have, again, one of those viral strains that starts the new infection. Then that can evolve, and then this individual infects this one, but you'll be able to track if you have samples from here, here, and here, these three right here, you'll be able to form a phylogenetic tree that shows you, yes, within this individual, there are three strains, and they all come from that same individual. But this one right here was also found in these other green individuals here, and this greenish blue one came from the first patient. So by forming these phylogenies, we can reconstruct the order of evolution of the different viral strains and the likely patterns of infection. So the blue individual infected the green individual that infected the orange individual. And so if you look at the real phylogeny that was used in this court case, there were two things that could have happened. The local controls, various individuals that had HIV in the population but weren't at that hospital and were not a patient of the doctor and was not the victim, obviously. The victim's name was Trahan. And if her blood sample was more embedded within a set of local controls, that means she got it in some other way. She didn't get it from him, not from one of his patients. However, what would happen if you get to something like this? All the local controls are more distantly related, and her viral strain is more closely related to one of his patients. Well, this is the real phylogeny, and what you can see is he had a number of patients that had HIV, and this is her viral strains, which she was already developing two different viral strains. But her HIV is clearly embedded within the clade of his patients, and the local controls are much more distant. So this was strong evidence that he actually did take a vial of tainted blood from one of his patients, which happened to be the patient at the base of this part of the clade. That was the patient that he had seen that day and taken a blood draw, and that's the same day that he gave Trahan a shot, which he said was a B12 shot, but it was tainted with his HIV blood. And so he was convicted. In the next video, we're going to continue our discussion of how we can use evolution to understand issues associated with human health.